Cephalopods. Is there anything they can't do? Let's look and see what they can and can't do. First of all, we've got these groups. We mentioned them before. So goniotites are most of what you're going to find around at the like in the Pennsylvanian, like in the times where we would have good fossil exposure here in Texas, in the Paleozoic at least. Ceratites are going to be in the early Mesozoic, and so you'll get some in the Pennsylvanian that have this sort of suture pattern that look as though they are trying to do something like that. But we don't have any genuine ceratites. We have lots of goniotites in the collection, and we have quite a few different kinds of ammonites. And what you can see here is that the suture line, so this is the line between the shells. This is the line that the little divider between the shells is taking. It almost turns into a fractal. It becomes very, very complex. And the idea is that the more complex this is, basically the more, I don't know if it's surface area, but like more linear length of this gap between the shells, where you can potentially force water in or potentially maybe force water out as a way to sort of accelerate the working of the siphuncle. Because diffusion is not a fast process, or maybe not fast enough if you're trying to chase something that's mobile. And if these are predators, which they seem to be, then they would have to be more mobile, a little more nimble than their prey, or else they're not going to make it. Now, pay close attention to these guys down here. You'd look at that and say, obviously, that's a gastropod, but they're big, and these are cephalopods. These are, yeah, pretty close to ammonites, actually. So these are going to be some more of those unusual shaped ammonites. All right, here's something you don't see every day, color patterns on early cephalopods. So here is a group of ammonites from the inimitable royal mapes, and they're photographing them in UV light. And so you can see these have like a color patch on the side, and some of these have real color sort of lines running up and down the body. Those are likely reflecting the color of the original shells. And it's been modified, like the pigments get fossilized the same as everything else. And so sometimes you can tell what a pigment probably used to be, what it looks like a derivative of if you do that sort of chemistry. And so you can reconstruct a few colors like black and red, for example. Other colors are trickier. And over here you can see some other cephalopod color patterns. Like, look at this, little scallops running around it, zigzags, fantastic. Stripes, very slimming. Oh my god, or maybe these are very slimming. Yeah, these are not. Yeah, but looks good. Looks really good. All right, so yeah. And because those shells are external shells, that would have been visible on the outside. And so they may have been able to see this in each other. It may have helped them to recognize who they should mate with, like who's in my species, who looks good. I have no idea what else these color patterns would have done for the organism, because they're pretty big predators. There are some larger, real big fish and shark predators at the time. They could have maybe crunched some of these things, so they're not quite the top predator, but they're up there. Yeah. Like there are still lions and tigers in the world, but we're up there. We do pretty well. Yeah. So here is looking at once again the position of the siphuncle on some of these organisms and the way that they are coiling. So you get some that are coiling in basically away from the eye. You get some of these that are coiling in toward the eye. You get some of these that are just straight, some of these that are mostly straight, but in their early growth, which is the little part, they are coiled up. Yeah, just quite a range. Some of these with a bloated sort of siphuncle, that is a thing that we will see in some of the fossils if you section some of the ones that we find here in Texas. All right. There's also the idea that as the shape of these is changing, it really changes what you can do with the shell. So with a shell like this, it is protecting your soft parts, so it's doing okay for that. But it is not much good for moving around, except in a straight up and straight down. Now they would have a siphon, so they'd be able to stick their siphon out. But imagine you're this tall and you're siphoning out here. You go like that in the water, and so they'd have to then sort of re-angle themselves, or they'd have to have some like way of holding their tentacles, or they'd have to try and swim at a certain angle so that they could move side to side. Side to side would have been difficult. Up and down would have worked just fine, but would have been dependent on flooding and unflooding some of those chambers. As we get these more curled shells, so this is the involution, like how much the outer whorls are going to wrap around the inner ones. This is going to give an increase in swimming velocity. They're going to get much better as swimmers as they start to do this, and potentially, you're going to get an increase in maneuverability here. All right, so if you've seen little dioramas like in Fine Art America's one right here with I don't know, that looks like a bunny rabbit face that AI put onto a trilobite. I don't know what's going on there. But you'll often see some of these cephalopods drawn out like this. And we're not saying that they never did this in life, but this would have been a thing that they could do for a little bit while they're traveling, and then they'd have to stop. Not least of all because if your siphon is pointed this way and your eyes are on the sides, then your shell is likely to run into things. And remember, these are organisms that we don't think had any way to repair damage to their shell. So if you start puncturing some of those chambers, that's like popping some of the watertight chambers in a submarine, and that's not good. It's not good for the sub, it's not good for the cephalopod. But if you do functional tests, as you should, so these are 3D printed models of some early nautiloid shells, and these are some nautiloid shells with sort of different amounts of curvature in them. What you find is when you put these things in water, they float just like this. That is their natural floating position. If there's a little bit of air in the chambers, it's going to make the organism float like this. So when you see them like this, either they were moving real fast, like they were scared of something, which, oh my gosh, what's over at that side? The picture, yeah, must be something real bad. Although these little guys are just going straight toward it, so I don't know. But probably more like this would have been the comfortable life position. And from there, it could, drop down. Now, you can look at human hands and say, human hands, clearly not, involved in swimming. They'd be rather poor at it. And that's fine. But, I do like swimming sometimes, and you may as well. You can do a lot of things. It's just we're looking at the adaptations and what would the natural advantages have been. I think that's all you can really infer from this. You can't absolutely say it never did this, it always did this, or rather those are very difficult things to be able to say. So, what happens if you float models or real shells of things like a nautilus? So here's a model of Ceratides, so this is going to be early Mesozoic. Here is a proper nautilus shell, and they put in a weight to simulate how much the organism itself would have weighed. And these things float in essentially the same body position that you will find the modern nautilus in. It's going to, be roughly looking like that. They were looking at some of the other types. This one is it has a lot of coils, a lot of chambers, and it's very like a very thin sort of Cinnabon. And those, if you leave them just unweighted and you don't have much water in the chambers, they float flat like this. And that would have been lethal to the organism. That would not have been a good look. So from this, they conclude that probably a nautilus or a ceratite didn't have to have as much water in its chambers while it was swimming, and you would have had to have a lot of water, essentially ballasting these chambers in order to get a shell like this with the organism at the bottom to actually sit in a sensible life position to where it could actually move around side to side and see what it's doing. Yeah. Because in this orientation, it'd have one eye pointed up into the sky, doing nothing, one eye pointed straight down, and it would only be able to spin around essentially on the surface with its siphon. Yeah, not a good look. Not how you survive in this world. Here's another kind of neat experiment from the same paper, Raymond. Actually, that may be French. I may be underpronouncing that. But this is showing they're putting some nautilus shells, or these are the ceratites actually, into a tank under a sort of a slow current. And so when these things go down, they bump against the bottom. And this is something that may happen because these nautiluses are not particularly strong swimmers because they're just moving by siphon, and they do their best, but they work best in a sort of a sheltered environment. But what's the shell going to do for them? Well, first thing, it's going to get pushed along like this. You see it gets pushed along straight, and then from about this point all the way to, you can see them right back here, it's going to turn sideways, and it's going to resist the current. And so that is just naturally what these shells are going to do. And so that's a benefit. If you're trying to scavenge something or hunt something that is in a particular spot, the shell will orient itself without you doing anything and will sort of resist that current and get pushed along rather slowly. All right. There have been a lot of functional studies lately on what does it mean, these different types of cephalopod shells. And so with some of these that are kind of narrow and have lots of little chambers, but they're very thin like this, they will have a lot of maneuverability. So they can really control whether they are swung back up here like this or like this. Just with a little toot on their siphon, they can turn down and grab something, because they can't just wait for the fish or whatever to, blunder into the correct side of them. They're not going to be a good predator if the prey has to do most of the work. Yeah. Unless they're an ambush predator, and I don't think they would be very good ambush predators. But over on this side, if you have a more involute shell, so this is an evolute shell where the subsequent layers don't overlap. The involute shells where the subsequent layers do overlap. This gives you a lot of stability, so it's harder for these things to rotate in the water. These things, it's easier. Just a little bit will send this rolling right around, and they've confirmed this with nice models. And then what you can see over here is, if you call this yaw, like their ability to tilt side to side, so not forward and back, but side tilt. There is a lot more maneuverability for the fat, chunky involute. And you would have a lot more stability, at least in terms of yaw, for the skinnier one. So, and there they've modeled it all with sort of simulated flow and the pressure that it would be putting on there. So yeah, kind of interesting what you can do. Certainly not the last we're going to hear of this. This is a pretty active field at the moment. All right. Here's another one, and so this one is looking at, all right, when these cephalopods are expanding their shells, and so here you can see the phragmocone, the body chamber, what are their options? So if they want to grow not that they want to, but if the body is going to grow bigger, then there are a couple ways they can expand the shell. So there are some of these where it seems to mostly expand shell width. And so you're going to get extra stability in one direction and extra instability in this direction. If you do that, you can increase the height of the whorls, but stay sort of thin. If you do that, then it's easier to tilt this way, front to back, harder to tilt side to side. They're going to be more resistant to that tilting. Or you could just try to generally increase the, you could even increase the length of the chamber. The phragmocone, and yeah, that essentially wouldn't change the hydrodynamic properties of the shell. So it's not that they were thinking in these terms and doing all the equations in physics. It's that when you had these and they did grow, these are the consequences of different types of growth. And so natural selection is going to be acting on these. If one of these makes you a little bit better at surviving in your ecological niche, hunting whatever it is you're hunting that stupid little preapulid worm that our little plectronocerises are trying to hunt, then they would thrive. And if you were good at something else, but it wasn't the thing that you're doing right now, they would either change environments or just die out. Yeah. All right, and the last part, let's talk about that dying out, shall we? All right, one reason to eat your greens is that when you do, plants are very good at bringing in certain isotopes of carbon. And so the more plants you eat, or the closer you are to something that's eating plants. So if you're eating a thing that is eating plants, eating a herbivore is the next best thing to eating plants in terms of the amount of carbon selection that you're getting. And if you're eating let's see, if somebody's eating a plant and then there's somebody that's eating the animal that's eating the plant and somebody that's eating that, and you have a big tall sort of pyramid and you're right at the top of it, apex predator. Or if you're scavenging dead things, or if you're mostly making your shell by just picking up atoms that you find in the water, then you're not going to get that carbon selection. And so the percentage of metabolic carbon is going to be the percentage of certain carbon isotopes that are going to be enriched in through eating other organisms, basically. And so you can calculate you can kind of estimate the metabolism of some of these ancient organisms based on the types of carbon that they preserve in their shells. So is it mostly carbon that's coming from eating other organisms? In that case, this is a thing that is probably growing fast. It's probably predation all the way. And it's one of these live fast, die young sorts of things. If you've got most of your carbon just coming from the environment, very little from actual hunting, then that would be indicative of potentially you're doing less hunting, so there's probably less energy available to you, so you're going to have a lower metabolism. That's the inference from this type of study, which is kind of an interesting way to look at it, and this is something you can measure from the shells of the animals. So, shown here, these are the shells of animals that still exist, so we can tell what they eat because we can just watch them, or put them in a tank. What would you like to eat? And so here you can see you've got some of these coleoids that have a gladius, like an internal sort of part, and those internal shell part, those are very active, and those are very predatory, high metabolism, high degree of hunting. There are these other different type of internally shelled coleoids, much lower metabolism that we know. Octopus, about in the middle, and in this case it doesn't have a shell of its own, but if it's something like an Argonaut octopus that actually does make an egg case, then yeah, you can grind up that egg case, and that's going to be analogous to the shell, and so you'd be able to tell the metabolic fraction from that. Nautiluses down here, they're scavengers. They are very slow, very low metabolism. They do not do a lot, but they don't have to, do they? They're just magnificent as they are. So those are things that exist. What about things that no longer exist? And what can that tell us about them? Here you go. What we've got here are some, and this is at different times, we've got some different organisms. And so there's different ways of estimating the metabolic carbon from these things, but you've got your ammonoids, so things like an ammonite, and you've got your nautiloids, things like a nautilus. And so you can see the nautiloids in the Maastrichtian, which I think is some part of the Mesozoic, pretty low. And with the ammonoids, you've got a mix. You've got a couple of them that are low, but most of them very high metabolism comparatively. Same over here in the early Maastrichtian, and these are all going to be fossils from the same formation, same environment, so it's a fair comparison. So they should have undergone the same amount of diagenetic change, like change during fossilization. Nautilus low and the ammonoid high. Yeah. So, based on this, you can say, yeah, ammonoids were probably high metabolism, highly hunting dependent, highly food source dependent creatures. And when their food sources were abundant, they probably did really well. And when there were extinction events that hit little things, even like the little single celled plants and algae, then those are going to ripple out because those are going to make it hard for the animals that eat algae in order to live. And those are the animals that are going to be eaten by the things that ammonites eat. And so if you knock out the bottom of this sort of food pyramid or chain, however you want to think it, everything else just collapses. And that's what happens in some of these major extinction events. So yeah, there's some of the evidence for why we don't have ammonites. We had a really long, really hungry time, and they were just not built to withstand long periods with very little live food that they can eat. Whereas nautiluses are happy with that today, and we have selected out, got rid of any nautiluses that may have been sort of high metabolism. Those did not make it through several extinctions. And so, yeah, what we've got is what was selected. And what we don't have is what was selected against. So there you go. That's our little evolution demo, and that has been cephalopods. Hope you found it interesting. I think they're a fascinating group. Thanks very much.